So the next few slides will look at the process of transduction, and this occurs when a bacteriophage transfers bacterial DNA from one bacterium to another during sequential infections. And there are two types, generalized and specialized. So during the lytic cycle, when the phage has been assembled, sometimes instead of packaging the viral DNA like they should, a phage can take a random piece of host DNA and insert it into its capsid. It will then inject the former host DNA into a new host. Then when DNA recombination occurs, this provides the new host with new genes, for example, an antibiotic resistance gene or a sugar metabolizing gene. So this slide looks at the process of transduction in more detail. So generalized transduction most often occurs during the lytic cycle of virulent phages, but sometimes happens during the lytic cycle of temperate phages. Any part of the bacterial genome can be transferred after being partially degraded as the virus takes control of its host. During the assembly stage, viral genomes are packaged by what's called head-full mechanism. That is, only genomes of a certain size are packaged. During generalized transduction, a fragment of the host genome that happens to be about the same size as the phage genome is mistakenly packaged. So specialized transduction is different, as this occurs at the end of the lysogenic cycle. During excision, a phage may occasionally remove some bacterial DNA near the site of viral integration and integrate it into the host. So as we said previously, DNA can recombine with the host, giving new characteristics. And transduction seems to play an important role in the evolutionary process of bacteria, giving them a mechanism for asexual exchange of genetic information. So this slide shows the process of specialized transduction in more detail. As we said, specialized transduction occurs at the end of the lysogenic cycle. So if we look at step 3, the excision step, here the phage DNA is excised from the bacterial chromosome, along with a short piece of bacterial DNA. The DNA is then packaged into newly formed capsids. Then the phage containing both viral and bacterial DNA infects a new host cell. This is seen in step 4. Then the last stage, stage 5, recombination, the phage DNA along with the attached bacterial DNA are incorporated into the new cell. So now we'll move on to the life cycle of animal viruses. Viruses that infect animal cells replicate by the lytic life cycle, even though not all viruses cause lysis of their host cell during replication. So the lytic animal viruses follow similar infection stages to bacteriophages, including attachment, penetration, biosynthesis, maturation and release. However, mechanisms of penetration, nucleic acid biosynthesis and release differ between bacterial and animal viruses. After binding to host receptors, animal viruses enter through endocytosis, which is engulfment by the host cell, or through membrane fusion. Many of the animal viruses, are host-specific, so that means they only infect a certain type of host. And most viruses only infect certain types of cells within tissues. And this specificity is called tissue tropism. So examples of this are shown by the poliovirus, which exhibits tropism for the tissues of the brain and spinal cord, or the influenza virus, which has a primary tropism for the respiratory tract. So this slide looks at the lytic cycle of the influenza virus. In the first step, the viral glycoproteins attach the virus to a host epithelial cell. Then in step 2, penetration, the cell engulfs the virus by endocytosis. Once the virus is in the cell, it uncoats, so this means that the genome is completely released from the capsid. This is step 3. Then in step 4, biosynthesis, the viral RNA enters the nucleus where it is replicated. And then new viral particles are assembled in step 5. The last step is the release of new viral particles, or virions, via budding. So budding results in the pinching off of the virion from the host membrane, thereby releasing the virion into the extracellular fluid. The cell, which is not killed in the process, then continues to make new virus. Just a note about this diagram, so on step 5, phage should read viral. So it should be the new viral particles are assembled on the diagram. So animal viruses do not always express their genes using the normal flow of genetic information, which is from DNA to RNA to protein. Some viruses have a double-stranded DNA genome, and they can follow this normal flow. However, many have single-stranded DNA, double-stranded RNA, or single-stranded RNA genomes. So if a genome is single-stranded DNA, host enzymes will be used to synthesize a second strand that is complementary to the genome strand, therefore producing double-stranded DNA. The double-stranded DNA then can be replicated, transcribed, and translated similar to host DNA. If the viral genome is RNA, however, a different mechanism must be used. If the virus possesses positive single-strand RNA, then the RNA can be used directly as mRNA and be translated into structural protein. If the virus contains negative single-strand RNA, the host ribosomes cannot translate it until the negative single-strand RNA has been converted into positive strand RNA by viral RNA-dependent RNA polymerase, RDRP. If we look at HIV, however, this follows a different pattern. So it's what we call a retrovirus and has positive single-stranded RNA, but this cannot act as mRNA. So this is transcribed into DNA by reverse transcriptase and incorporated into host DNA. The subsequent transcription to make mRNA and viral genomic RNA is under the control of host transcriptase enzymes. So viruses can be grown in vivo or in vitro. Bacteriophages can be grown using bacteria culture, and lytic phages can be observed as clear zones, whereas animal viruses require cells within a host animal or tissue culture cells derived from an animal. Cultivation is really important for identifying and diagnosing viral disease, also for the production of vaccines and for basic research purposes. In vivo host sources include the developing embryo in a bird's egg. So, for example, flu vaccines are cultured in hen's eggs. Primary cell culture can also be used for viruses. So the next few slides look at methods for detecting viruses. So one method is to look for certain cytopathic effects. So if you take a sample from the infected host and look at this under a microscope, for example an electron microscope, you will be able to see the cell abnormalities due to viral infection. So CPEs, cytopathic effects, can include loss of adherence to the surface of the container, changes in the cell shape from flat to round, shrinkage of the nucleus, and vacuoles in the cytoplasm. The type or severity of the CPE depends on the type of virus involved. So another method of viral detection is the hemagglutination assay. And this assay can be used to detect specific types of viruses in the patient's serum. So hemagglutination is the clumping together of red blood cells. Many viruses have spikes called hemagglutinins, and these can bind to receptors on the membranes of red blood cells, causing the red blood cells to agglutinate or clump together. This hemagglutination is observable without using a microscope. To identify a specific pathogenic virus using the hemagglutination assay, an indirect approach is used. So virus-specific antibodies can be used to bind to the hemagglutinins found on the virus. This then prevents the red blood cells from directly interacting with the virus, so agglutination is inhibited. So the table on the slide shows the possible outcomes of the hemagglutination assay. A shows the case if no virus is present, so the red blood cells do not bind together and they sink to the bottom of the well, and this is visible as a red dot. Then B shows the virus and the red blood cells together, and it shows the agglutination, the clumping of the red blood cells, due to the hemagglutinin present on the virus. And you can see that this results in the red colour throughout the well. C then shows that when virus-specific antibody, virus and red blood cells are added together, the antibodies inhibit agglutination and can be seen as a red dot at the bottom of the well. So next we'll talk about PCR and RT-PCR. So these are nucleic acid amplification tests and are used to detect unique nucleic acid sequences of viruses in patient samples. So PCR, or polymerase chain reaction, is used to detect the presence of viral DNA in a patient's tissue or body fluid sample. PCR is a technique that amplifies, so it synthesizes many copies of a viral DNA segment of interest. Using PCR, short nucleotide sequences called primers bind to specific sequences of viral DNA, enabling identification of the virus. And then we have reverse transcriptase PCR, RT-PCR, and this is used to detect the presence of RNA viruses. So RT-PCR differs from PCR in that the enzyme is reverse transcriptase, and this is used to make a complementary DNA from the small amount of viral RNA in the specimen. The complementary DNA can then be amplified by PCR. Both PCR and RT-PCR are used to detect and confirm the presence of the viral nucleic acid in patient specimens. Enzyme amino acids can be used to detect viruses, and this requires a few drops of diluted patient serum or plasma applied to a membrane filter. Then the next step is to add antibody with enzyme conjugate. Okay, so this is antibody conjugated to an enzyme. Then the antibody will attach to any viral antigen present. Then any unattached conjugate is washed off, and then the substrate is added. So in a positive sample that contains virus, the substrate and enzyme interaction creates a colour change for detection. However, in a negative sample, there's no colour reaction due to lack of enzyme, okay, because the enzyme has just been washed off. So we'll just talk briefly about virus-like particles. So viroids are acellular virus-like particles, but they consist only of a short strand of circular RNA capable of self-replication. The first viroid was found to cause potato tuber spindle disease. Like viruses, viroids take control of the host machinery to replicate their RNA genome. But however, unlike viruses, viroids don't have a protein coat to protect their genome. So these viroids, they mostly affect agricultural crops. So virusoids are non-self-replicating single-stranded RNAs. And these belong to a large group of infectious agents called satellite RNAs. RNA replication with the virusoids is similar to that of viroids, but they require helper viruses. Then once helper virus enters the host cell, virusoids are released. Virusoid genome does not code for any proteins, but instead serves only to replicate virusoid RNA. One example is hepatitis delta virus, and the helper virus for this virusoid is the hepatitis B virus, or HBV. And it's found that co-infection with HBV and HDV results in more severe pathological changes in the liver during infection. So prions are proteinaceous infectious particles, first identified in 1982. They don't contain any DNA or RNA or genetic material. Prion diseases are also called transmissible spongiform encephalopathies, or TSCs, and these are fatal neurodegenerative disorders that have attracted enormous attention, not only for their unique biological features, but also for their impact on public health. Prions consist of abnormally folded proteins that induce normal forms of the protein to fold abnormally. This group of diseases includes Scrapie-Kuru, Creutzfeldt-Jakob disease, and variant Creutzfeldt-Jakob disease. Kuru was a disease seen in Papua New Guinea among people who ate human tissue, including brain. Variant Creutzfeldt-Jakob disease, VCJD, arose after animal brain protein was fed to cattle, the disease being transmitted to humans by ingestion of contaminated beef products. The animal disease bovine spongiform encephalopathy which is BSE, has been eliminated in countries with effective control measures. The size and scale of any human epidemic is uncertain but likely to be small. So in summary, viruses are living molecules. They're obligate parasites. They are host and cell specific. They also have unique shapes and structures that enable them to infect target cells in a specific manner. And different phages and animalviruses have different life cycles, and their life cycles play a role in infection and control of host cell function to induce viral diseases. Isolation, cultivation, and detection of viruses are very important for the diagnosis and treatment of viral diseases. And then we also have the virus-like particles, so the viroids, virusoids, and prions, which also play key roles in infection.